Welcome everyone to the Vitality Equation, exploring research in health and well-being one paper at a time. Today, we're diving into a fascinating, albeit dense piece of research about lung adenocarcinoma, a type of lung cancer. It's titled SIRT2 TFEB CNI H3ADRM1 NTM9 SF4 as potential B IOMARKER S of resistance to CRI ZOTI NIB and poor prognosis in lung adenocarcinoma. Implications for targeted therapy in IMMUNOTHERAPY. So quite a mouthful.
A mouthful indeed. But underneath that title lies potentially crucial information for improving treatment outcomes. I guess the core takeaway is that this paper identifies specific proteins, these biomarkers, SIR, T2, TFE, and the others, that seem to indicate whether a patient with this type of lung cancer is likely to respond well to a particular drug called Crizotinib.
Right, and poor prognosis is never something you want to see associated with your biomarkers. So if these proteins are present or behaving in a certain way, it might signal that the cancer is less likely to respond to the drug, and unfortunately, the outlook for the patient might not be as good.
Exactly. And that's where the importance of this research really shines. Think about it. If we can identify these biomarkers early on, we can potentially avoid wasting valuable time and resources on a treatment that's unlikely to work. That time could be used to explore other options, maybe different targeted therapies or even immunotherapy.
Yes, and it also opens up the door for more personalized medicine. Instead of a one-size-fits-all approach, we can tailor treatment strategies based on an individual's unique biomarker profile. It's like understanding the specific wiring of someone's cognitive biases before designing a personalized CBT program.
I like that analogy. It's like knowing whether someone's a marathon runner or a sprinter before designing their training program. You wouldn't put them on the same regimen, right? Knowing these biomarkers is like understanding the patient's cancer fitness level, so to speak.
Okay, let's break down some of these biomarkers. SIRT2, for example. It's a protein involved in various cellular processes, including aging, DNA repair, and inflammation. From my understanding, and correct me if I'm wrong, increased SIRT2 activity is linked to crizotinib resistance in this specific type of lung cancer.
That's my understanding as well. It's almost counterintuitive because SIRT2 is often associated with positive health outcomes in other contexts. But in this specific cancer, it seems to be helping the cancer cells survive and resist the effects of the drug. It's like sometimes cognitive flexibility, which is usually beneficial, can also enable someone to rationalize harmful behaviors.
That's a really interesting point. It's like how too much exercise can actually be detrimental, leading to overt raining and injuries. Balance is key, even at the cellular level, it seems. So if SIR-T2 is elevated, does that mean we should be looking at therapies that specifically target and inhibit its activity in these cases?
That's definitely one avenue to explore. The paper suggests that targeting SIRT2 could potentially resensitize the cancer cells to crizotinib, making the treatment more effective. But it's not as simple as just turning it off completely. We need to understand the downstream effects and potential side effects of such an intervention.
Exactly. It's like trying to rewire someone's thought patterns. You can't just delete a thought, you need to replace it with a healthier one. Similarly, you can't just block SIR-T2 without considering what other processes it might be affecting. What about TFB? What role does that biomarker play?
TFEB, from my reading, seems to be a master regulator of autophagy, which is essentially the cell's self-cleaning process. It helps the cell break down and remove damaged or unnecessary components. In this context, it seems that increased TFEB activity is also associated with crizotinib resistance.
So if the cancer cells are better at cleaning themselves up and removing damaged components, they're potentially more resilient to the drug's effects. It's like optimizing cellular efficiency for survival, even in the face of treatment.
That's a great way to put it. It's like a super efficient cognitive defense mechanism. The cancer cells are essentially clearing out the damage caused by the drug, allowing them to survive and proliferate. Could this autophagy process be something we could target with cognitive techniques?
Perhaps. But in terms of physical health, it makes me think about the importance of sleep and recovery. When we sleep, our bodies are essentially engaging in their own form of autophagy, cleaning up cellular debris and repairing damage. Could optimizing sleep potentially enhance the effectiveness of cancer treatments by supporting this natural self-cleaning process?
That's a fascinating connection. So better sleep could, in a way, prime the body to respond better to treatment by optimizing the cellular environment. Maybe some of my guided meditations that focus on deep relaxation and cellular repair could be helpful.
That's an interesting idea. And linking that back to cognitive processes, perhaps mindful meditation could help regulate the body's stress response, reducing inflammation and further supporting the cellular environment. We need to find the right balance. Is there a way to measure it?
Absolutely. There's definitely emerging research on the link between stress, inflammation, and cancer progression. So, managing stress through meditation, exercise, and proper sleep could potentially play a role in improving treatment outcomes. Let's talk about CNIH3, M1 and TM9, SF4 now. What are their roles?
Okay, so CNI, H3A, DRM1, and TM9, SF4 are a bit less well known, at least to me. From what I gathered, CNI, H3 is involved in receptor trafficking, which basically means it helps move proteins around within the cell. ADRM1 is a proteasome subunit, which is part of the cell's protein degradation machinery, kind of like a garbage disposal system. And TM9, SF4 is a transmembrane protein involved in cellular transport.
Right, so it sounds like all three are involved in essential cellular processes. And if these processes are disrupted or altered in some way, it could contribute to crizotinib resistance. Thinking cognitively, it's like a disruption in the communication pathways within the brain, leading to cognitive dysfunction.
Yeah, it's like a breakdown in the cell's internal communication and waste management systems. If the proteins aren't being transported correctly, or if damaged proteins aren't being disposed of properly, it could create an environment that favors cancer cell survival. Perhaps resistance training can encourage proteins to act as they are designed.
So the million-dollar question is, how can we use this information to improve patient outcomes? We've talked about potentially targeting SIRT2 and autophagy, but what about these other biomarkers? Could we develop drugs or therapies that specifically target CNIH3, ADRM1, or TM9, SF4?
That's definitely a possibility. But it's important to remember that these are just potential biomarkers. We need more research to validate these findings and to understand the precise mechanisms by which these proteins contribute to crizotinib resistance. It could potentially involve looking at cognitive training exercises.
Exactly. Correlation doesn't equal causation. Just because these biomarkers are associated with poor prognosis doesn't necessarily mean that they're the direct cause of the resistance. There could be other factors at play. I wonder how this applies to exercise.
Right. What about combining targeted therapies with immunotherapy? Could we use these biomarkers to identify patients who are more likely to respond to immunotherapy? This could boost a patient's immune system to fight the cancer and maybe combine it with lifestyle changes.
That's a great point. Immunotherapy is a rapidly evolving field, and it's shown remarkable success in treating certain types of cancer. But it doesn't work for everyone. So, if we can identify biomarkers that predict response to immunotherapy, we can potentially save patients from unnecessary side effects and ensure that they receive the most effective treatment. Could we add CBT techniques into this mix?
Yes, and what about exercise? There's growing evidence that exercise can boost the immune system and improve the effectiveness of immunotherapy. Could we use these biomarkers to tailor exercise programs for cancer patients, optimizing their immune response and improving their overall prognosis? I guess the question is, are there personalized exercises for each person?
I love that idea. It's all about taking a holistic approach to cancer treatment, combining targeted therapies, immunotherapy, and lifestyle interventions to create a personalized treatment plan that addresses the individual's unique needs and circumstances. I believe that it is a more ethical way to ensure that all options are available.
Absolutely. And it's not just about extending lifespan, it's about improving quality of life. By managing side effects and optimizing physical and mental well-being, we can help patients live fuller, more meaningful lives, even in the face of cancer. And again, CBT techniques can influence that.
Yes, and that brings me back to the importance of sleep. Cancer treatment can be incredibly disruptive to sleep patterns. And poor sleep can worsen side effects, weaken the immune system, and negatively impact quality of life. So, prioritizing sleep hygiene and addressing sleep disturbances should be an integral part of cancer care. So that's another important element to consider.
Definitely. And that's where mindfulness and meditation can come in. They can help patients manage stress, reduce anxiety, and improve sleep quality, ultimately enhancing their overall well-being and potentially improving their response to treatment. Thinking slow and deliberate helps in so many situations.
That reminds me of something I read about seed dispersal strategies of Solanaceae might lead to new insights into optimizing sleep patterns. Could synchronized rhythms like the plant seed dispersal relate to a more effective sleep cycle and the regeneration aspects of sleep?
Speaking of seeds, I learned that specific environmental conditions have played a crucial role in shaping plant evolution. That made me think about how my daily environment and lifestyle choices directly influence my own health and well-being. It also sparked an idea for new guided meditations focused on the resilience of nature.
Okay, stepping back to the biomarkers, it's important to remember that this research is still in its early stages. These are just potential biomarkers, and more research is needed to validate these findings and to determine their clinical utility.
Absolutely. We need larger studies to confirm these findings and to assess the predictive value of these biomarkers in different patient populations. It's also important to consider the cost and feasibility of implementing these biomarker tests in clinical practice.
And we need to explore the underlying mechanisms by which these biomarkers contribute to crizotinib resistance. Understanding these mechanisms could lead to the development of new and more effective targeted therapies. I find the link to nature fascinating.
Yes, and it's also important to consider the ethical implications of using these biomarkers. We need to ensure that these tests are used responsibly and that patients are fully informed about the potential benefits and risks. This is going to require new studies.
Agreed. And we need to address potential disparities in access to these tests. We need to ensure that all patients, regardless of their socioeconomic status or geographic location, have access to the best possible care. So many factors to consider.
So let's talk a little bit about future research directions. From my perspective, I think it would be fascinating to explore the potential of lifestyle interventions like diet, exercise, and mindfulness to modulate the expression of these biomarkers. Could we potentially reverse crizotinib resistance through lifestyle changes? Could more study on how the mind influences biomarker expression be helpful?
From a cognitive perspective, I'd like to see more research on the role of stress and psychological well-being in cancer progression and treatment response. Could cognitive behavioral therapy or other psychological interventions improve outcomes for patients with lung adenocarcinoma? There is not a lot of work in this area.
And from a physical health perspective, I'd like to see more research on the impact of exercise and sleep on immune function and treatment efficacy in cancer patients. Could personalized exercise and sleep programs improve outcomes for patients with lung adenocarcinoma? I have seen a lot more studies showing the mind and the body are closely linked and should not be treated as mutually exclusive.
I think a collaborative approach, bringing together experts in oncology, genetics, lifestyle medicine, and psychology, is essential to making meaningful progress in this area. We need to break down the silos and work together to develop more effective and holistic approaches to cancer care. Perhaps some plant-based life hacks could play a role.
Yes, and we need to involve patients in the research process. Their experiences and perspectives are invaluable, and they can help us design studies that are more relevant and meaningful to their lives. If we can understand the cognitive elements, there may be a potential application.
Absolutely. It's about empowering patients to take control of their health and to be active participants in their own care. It's about giving them the tools and resources they need to thrive, even in the face of cancer. The whole team has to work together.
Well, this has been a really insightful discussion. It's clear that there's still a lot we don't know about lung adenocarcinoma and crizotinib resistance, but research like this gives us hope that we can continue to make progress in the fight against this disease. We should also be mindful of the techniques used to cope with the stresses of a diagnosis.
Yes, and it highlights the importance of personalized medicine and taking a holistic approach to cancer care. It's not just about treating the cancer, it's about treating the whole person. And if we can influence even one person with our discussion, that's a victory.
Definitely. By combining targeted therapies, immunotherapy, and lifestyle interventions, we can potentially improve outcomes and enhance the quality of life for patients with lung adenocarcinoma. There are also lots of studies that could be added to this area of research.
Thank you all for joining us on the Vitality Equation. We hope you found this discussion informative and inspiring. Until next time, take care and be well.